Okay, we're ready.
Welcome, everybody. You've arrived at the CyVerse Focus Forum webinar, and today Bonnie Hurwitz is presenting on iMicrobe. And so there's just a little bit of housekeeping to do. Please make sure you open your chat window, because that's where you'll need to type in any questions you have for Bonnie, and she'll either decide to answer them in her presentation or at the end, depending on how. We've got a bit of an echo, so I'm not sure if it's somebody on your side who needs to mute Bonnie.
Oh, yeah.
Okay. Thanks. And then in addition to opening your chat, in the chat I put in a wiki page where you'll find the information from today that we'll post later on. It'll take us a while to process the video properly, but you can see right there, and that's open, and you can come visit that wiki page anytime and rewatch the video and look at the slides. We'll also post the chat there. And my final bit of housekeeping is that we will have another focus forum in 2 weeks, and that'll be John Fauner from Texas Advanced Computing Center, and he'll be presenting on the CyVerse Software Developers Kit. So stay tuned for that. You can also find that event listed on our website, Cyverse.org. So without further ado, here's Bonnie Hurwitz.
Great. Can you all hear me? Okay, excellent. So welcome from iMicrobe headquarters. Today we're going to be talking about iMicrobe, but first I'd like to introduce you to two very important people on the project. The first is Ken Ewens-Clark, who is one of our core developers on the team, and also one of the data architects. And we also have Matt Baumhoff joining us here as well, who is another developer on the team. And we have Joshua Lynch, who is joining us live from New York as well. So thank you very much. And let's go ahead and get started. So, so let's see, let's go into, get my presenter mode up here. So today I'm gonna be telling you guys about the iMicrobe project and the work that we have done on really fostering community-driven science and data discovery. And all of this work is really bolstered by work with CyVerse and through Simas Foundation. And the Moore Foundation. Looks like we're on the wrong mode again here. So we're gonna swap displays real quick. Can you, nope, that's not working quite yet. Here, let's, can you guys see now? No, we're still in presenter mode here. Okay, we'll try this one more time. Use slideshow. Okay, nope, that's not working for us either. One more time. I did. Let's try use presenter view. Can you guys still see? Nope. We're working on getting you guys so you don't see my slides here as we go. So swap displays a little better. Moving forward. As it gets. Okay. Here, let's try. No, yeah, I guess I think you just go. We're just going to go with it. Okay. So. So one of the things that iMicrobe really, I'm going to move you guys down too. Can you see my screen? Oh, no, you can't. Okay. So with iMicrobe, one of the things that we're interested in doing is building a cyber infrastructure for microbial ecology. And as part of that, the iMicrobe project is really integrating different data types and analyses and making it possible for the community to use tools that are important for microbial ecology. All of that work is built upon CyVerse, where we have all of our data storage and compute. And in addition to that, the know-how and really the information about how to use all of these resources, we've been developing different protocols through protocols.io, and we're also really heavily involved in the Trellis forum, where we have active discussions about different different aspects of microbial ecology, all the way from tool development to the science behind it. So now it's not advancing. Let's see. There we go. So I just want to give you a little bit of an idea about where we got started. So originally, the iMicrobe project was born from a Gordon Betty Moore Foundation grant. And what happened was several years ago, the CAMERA project got started, and they worked on really providing this great resource for the metagenomic community, where you could store not only data sets, but also all of the metadata associated with the data, which is important for understanding really not just the microbes that are present in an environment, but also all of their physiochemical attributes, the location they were isolated from, and beyond. And that project went on for about 7 years, and it sunsetted in about 2014. And at that point, all of the data wasn't actually in all of the repositories, such as NCBI. And so what we did is we came along and we brought those data into CyVerse under a grant from the Gordon Betty Moore Foundation. And we started off with an initial platform that we developed in Perl Mojolicious. And this was a quick and easy way for us to be able to connect with the data sets in CyVerse and provide an interface for our users. And all of that was originally developed by Ken between 2014 and 2017. And then since then, we've actually moved to a brand new website that is all based on a programming language called Elm. And just to give you a quick reason why we did this sort of refactorization, there's a website that is currently called called redink.com, and that particular website was originally written in Java, and it had over 60,000 errors over the course of several years. But since they moved to Elm, it's actually had absolutely no runtime errors. And so we're moving our platform to that same kind of resilient web base, and allowing our users to have pretty much zero downtime as part of the website. And that new release happened in November of this past year as part of the National Microbiome Data Collaborative meeting. So I went to that meeting and I shared with various scientists there the work that we had done in iMicrobe. So really, we've made a huge amount of progress over a very small amount of time with just a few developers. Why is this not moving forward? Okay, there we go. So one of the aspects that's really important with iMicrobe is that we leverage the CyberCyber Infrastructure. And what that means is that we actually can store all of our data sets within CyVerse. And our users can also store their data sets and access them within CyVerse. And then all of the compute and processing engine for the applications that we have on our website that I'll tell you about shortly are all powered by CyVerse. And what this means is it gives users access to these high-performance computing resources that exist at Texas Advanced Center for Computing and also here at the University of Arizona. And then we access all of those different services using science APIs. And in particular, we're using the Agave API to pull data from the data store and then push it off to TAC for processing on the high performance computers. And just to give you, this is not moving forward here. So I'd like to give you just a quick tour of our interactive website. And so when you first come onto our website, the very first thing that you'll need to do is you need to have a CyVerse account. So you should have already registered for a CyVerse account through the CyVerse website. And once you have that CyVerse account, you can use iMicrobe as well as CyVerse for free. And here I'm going to show you just the simple process of logging in using CyVerse on our account on our site. So here I'm just scrolling through the iMicrobe site, and I can click on the login button, and then I can put my name in and sign in, and then I can go and I can look at my profile, and sure enough, it shows that I'm there and that I have actually logged in. So moving forward, iMicrobe, one of the important things about iMicrobe initially was that we could provide this data commons, and that data is all of the camera-based data sets that exist within the data store. And since then, we've also added existing additional data sets from viral ecology, as well as other data sets from the Hawaiian Ocean Time Series, and in particular, the Ohana Gene Catalog. So we're really trying to add data sets for the community that are of particular importance, or data sets that have been highly processed and actually offer an important resource to the community, as in the Ohana Gene Catalog data sets. And so what we do at iMicrobe is we can essentially go from the website through an HTTP and JSON to the RESTful Agave API, and then we can connect in with the data commons that way. So that's how we're actually accessing all of the data sets in CyVerse. So none of the data live on any of our iMicrobe servers. And so if we wanted to go and browse for data, we can do that on the iMicrobe data on the website. And once we have these data, we can also add those data to our cart for further processing. So let's take a look at what that looks like. So as we get started, we can go to the search engine, and I can type in Tara. So in particular, I'm looking for the Tara Oceans datasets. And here I'm looking for the Tara viromes. You can see I selected the project. I can also see all of the different samples that are associated with that project. And from there, I can see all the information and attributes that are actually associated with that sample data. And then I can view that particular sample on a map, and then zoom in or zoom out to see where it is on the map. And I can add all of those data sets into my cart for processing. So here you can see I've added 43 data sets into my cart. I can also remove data sets. So if I'm not interested in a subset of those, I can remove them. And then from there, I can go to add other data sets within from the site as well. So here I'm actually using our metadata search to search by a whole variety of parameters. And here I'm selecting the Longhurst province, and in particular, I wanted to add in data sets from Antarctica. So here you see me adding the viral metagenomes from Antarctica into my analysis, and you can see that they have gone into the cart as well. So now I'm ready to really move forward with processing data from the cart. So from there, the other added feature that we have on the iMicrobe site is that not only are the data sets available and that they're queryable within this metadata search, but we've also gone and indexed all of the data in terms of adding taxonomy and protein annotations to all of our data sets. And what that means is that we can go and search by your favorite organism or a particular protein of interest within the data sets. And so let's take a look at what that looks like. So here I'm going under Tools. I'm going to the taxonomy search. And something of interest is, hey, we have a microbial website, but hey, let's go look for anything that's human, because as we sequence all of these samples, we can certainly sprinkle in human samples or human DNA. And so we can see that some of the host microbe data sets are, in fact, have human reads, and we can filter those by abundance. So if we're only looking for data sets that seem to have a high amount of contamination, we can see those here under the taxonomy search. We can also search by specific proteins. So here I'm going to add in a PFAM ID for a protein of interest. And then I can go ahead and search for that particular protein. And I can also look for a particular abundance of reads. And here you can see I'm filtered for anything with a read count over 50 reads. So it's a really powerful way to be able to look for particular data sets, either through metadata searches, which are essentially searching across all of the metadata associated with the samples. So any of the physiochemical data can be searched that way, or we can also search by taxonomy or proteins that are associated with the actual sequences within the data sets that we have.
Bonnie?
So the other useful utility within the iMicrobe data, within the site, is that we can also download the data from iMicrobe. And there's several ways of doing that. We can either do it by downloading directly from CyVerse, where you're taking it off the data store, or we can do that by downloading the data from the FTP site directly. So let's take a look at what that looks like. So here I have a number of data sets in my cart. I can see those files. I can search by types. So here I'm saying I only want the reads that are associated with the data in my cart. And I can click on one of those and I can go to CyVerse and I can download those reads directly. The other option is I can go to download, and you'll see that our entire FTP site is shown here for you to access and download. So if you're a programmer, you can download these data sets directly from the FTP site, or you can download them online as well. So moving forward, one of the other really amazing aspects associated with iMicrobe is that because we're integrated with CyVerse, we're able to run applications directly from our site. So what we're doing is we have our own applications that we have developed from various tools, and we also have containers, the entire suite of applications that are public at CyVerse that we can add to our site. Or we can add tools that are coming from biocontainers. And so I'll tell you a little bit about all of those aspects. So here, what we're doing essentially is we're going through the Agave API, and we're pushing data across to the compute systems over at Texas Advanced Center for Computing. And then we can run different applications on those high-performance computing resources. So what's under the hood? We just recently moved all of our applications over to Stampede 2. Stampede 2 is one of the largest clusters the 12th largest cluster in the world. It has approximately 18 petaflops at its peak, and it was built recently, given a $30 million award from the National Science Foundation. So it's an incredible resource that powers all of the applications on our site. So here we go. So how do you run an application from iMicrobe? So one of the great things is that we've just gone through showing you how to add data into the cart. From there, we can actually take all of those data sets and we can then run them. We can select certain components of them and run them through an application on our site. So here what we're doing is we are looking within our cart. We go down to the tools and to applications. You can scroll through the list of applications and you can find your application of interest. I'm selecting the MASH all versus all. And then here I've gone to my cart and I've selected different data sets that I want to include or exclude within the cart. I am also selecting the kind of data I want, which are reads for this particular application. I can change the parameters, and then I can submit the job. And then you can see that it's running, and you can see it's staging the inputs and what it's actually doing. So there's a lot of communication about what's happening on the site at the time. And then you wait for a little bit of time, usually not terribly long, but it depends on the application and what you've submitted. And then you see a status of finished, and you can see all of the information about it. And in addition, we've added these interactive D3 visualizations of the data. So I can not only see the results of my data, but I can actually see those interactive maps. Here I'm logging into CyVerse, and I can go through and actually access all of those files directly in CyVerse. From there, I can share the files with other CyVerse users, or I can download them to my laptop for further analysis. Or you can use iRods to shuttle them from CyVerse to a server that is running CyVerse or running iRods as well. So really a huge amount of things that you can do and run here. And so one of the other things that you might want to do is, after you have run a job, let's say you decided you want to go and get a coffee, and you're going to come back and you want to see how your job is doing. And you want to take a look at the job history. So here we're going to show you how to actually access the job history. So if I, oops, here we go. So I can go to Tools again. I can go to Jobs. And from Jobs, you'll see all of the jobs that have been run. And you can see their status. So here we see some are running, some are archiving, some are finished. I can click the MASH job that I've just run. And I can see that it's finished. I can see the details about the inputs, the parameters, the outputs. And again, so you can access those visualizations at any time, or you can also go off to CyVerse, as I had shown you earlier. So a lot of flexibility for running jobs. So the other question that you might have is, okay, you have all of this great data within iMicrobe, but how do I run my own data through an app using iMicrobe? So let's take a look at that. So the very first thing we're going to need to do is we're going to actually need to put your data set into CyVerse, into the CyVerse data store. Oops, here we go. So I'm going to log into CyVerse. And after I've logged in, it'll take me to the discovery environment. And I click on that and I launch it. And then once the discovery environment has loaded, I can click on my home directory and I can go to a particular data set, let's say webinar, and I can upload a sample and add it from my home directory or my laptop into my CyVerse account. Here I'm adding a couple different query sequences in there from my laptop. And then I can upload those, and then you'll see those going into my CyVerse directory. And now I have a number of SRR files ready to go for the analysis. And then from there, I can go to iMicrobe. I can log in. Again, I'm authenticating with my CyVerse ID. And from there, I can go to Tools. I can go to Apps. And I can select a particular app. In this case, I'm running Centrifuge, which is an application for looking for the taxonomic identity of different microbes in those data sets. I can go to the webinar folder and select that. And then I can click on Run in order to, or change the database or any different parameters I want. And then I can run it, and then that submits my job. And you'll see again, this will take a little bit of time to kick off. My job is now pending. And from there, I can wait for that to finish. And you'll see a little bit of time passing here until it's finished. And you'll see a little time later. And there we go. Look, my job is finished, and I'm ready to take a look at the outputs. Here are my output files. And all of those outputs, again, are in the CyVerse data store for you to download or share. But we've also got them visualized here on the site as a bubble chart. So you can see these samples were ocean samples, so there was a lot of Prochlorococcus. Or I can go back to CyVerse, and here I am going to my analysis folder and my jobs, and I can share or download those files directly with anybody such as my PI or other collaborators working on the project. So one of the things that is incredibly exciting that we're doing currently is we are starting to connect with different, so we have a number of tools that we have developed on our own through iMicrobe. But we also realize that the community is developing in a wealth of different tools through biocontainers. And if you don't know what a biocontainer is, it's essentially a virtual machine where there's an operating system, there are dependencies, and then there is code that is all embedded in a single unit. And what's fantastic about biocontainers or containers in general is that you're able to run those containers on any server anywhere, and all of everything that you need is within that container. And so we use those containers to run on Stampede too, and execute our jobs that way. And recently we started bringing in containers that have been developed from the community. And so what has recently been done at TAC is they've actually brought the capability for downloading those biocontainers directly from Stampede. And so we've just written some of the glue that lets us create a that application-based web form that you see with all of the parameters and submission, and then we use that and then we can tie that with the Agave API, and then run them on Stampede too. And so what this gives us is the capability to really bring any biocontainer in. And so what we're asking the community is, first of all, are there particular biocontainers that you would like to see in iMicrobe? And please, tweet us. Let us know what you're interested in. And then secondly, if you have a favorite application that isn't available currently as a biocontainer, CyVerse offers container camps that you can learn how to take different bits of code and containerize it, and then let us know about it, and we can start to make those containers available to the community as well. So one of the things that we're particularly interested in through the iMicrobe project is really interfacing with the community, making these applications available, because what that enables us to do as a community is to look at these tools side by side in a single, easy to run web-based format where we can compare and contrast the results and maybe have a lot of fun doing some assemblathons or deciding what are the best gene callers, and doing that all within a single environment where the compute is connected and readily accessible to anybody from any realm of computing. So another aspect is that we're really very interested in helping the community to establish protocols and workflows. So as various innovations happen in the computational world, it seems like there's an endless number of tools, but really there's everybody has their secret sauce for how they might create an assembly or do various aspects. And so what we've been doing is taking some of those recipes and then putting them into protocols.io where they can be reused by the community. And what's fantastic about protocols.io is people can actually comment on them. They can get forked, they can get reused in different ways. So you can add your own additions to those different protocols. And all of that has provenance back to the original protocol. So people can get credit for their work, both in developing code and in developing protocols. In addition, we've started to really interface a lot more using the Trellis communities, and that's a nice way to have very active discussions, both in the scientific and computing realm. And then one of the aspects that I really want to share with you too is that we are able to bring in applications from different communities, and we can actually tag them as being from a particular community. So here, we've actually brought in a bunch of applications that originally were written by Ben Boldock and as part of the Sullivan and Rich Labs at Ohio State. And this is part of the iVirus project. So these are all applications that are particularly important for analyzing viral data sets, either viral genomes or viromes. And so it makes all of those different applications available, accessible, findable discoverable within the iMicrobe website. So this kind of work really helps us to fuel those communities. And then, in addition to that, from a data standpoint, my group was recently funded for Planet Microbe, and what we're doing there is we're very interested in oceanographic data sets. And one of the things that tends to happen is that once a cruise goes out and the bottles come up from the deep darks depths of the ocean, those containers go to different labs for processing. Some go for looking at the chemistry, others go to omics analyses, and what we're doing within Planet Microbe is we're trying to bring all of those biological and chemical data sets back together along with the omics data so that we can really use all of those data sets in conjunction with one another. And we're starting initially with data from the Hawaiian Ocean Time Series and Bermuda Atlantic Time Series. So all of those data sets will be coming online in iMicrobe. And then we're adding in various NSF Science Technology Centers. And then from there, we're linking with other EarthCube efforts that are that have other important ocean data sets as well that we can link with these data. So really building not just a universe of tools, but also a universe of very nicely connected data sets for this community in oceanography. And this work was funded by the National Science Foundation. And then in addition to that, in other work that we're doing through the National Science Foundation, we have been working on developing algorithms for doing massive comparative metagenomics. And that uses a brand new kind of infrastructure called Hadoop, which is actually not yet available at CyVerse. But we have attached our own Hadoop cluster onto the iMicrobe, servers, or onto the iMicrobe service infrastructure. And we are using a particular kind of algorithm that we developed called Libra. And what that allows us to do is a massive all versus all analysis of sequence data. And here what I'm showing you is our 48 different human microbiome data sets from diverse body sites. And what we can see is that when we do a comparison with an algorithm such as MASH, which does sketching, what we see is kind of limited resolution, but nice separation on the data sets with both 16S and whole genome shotgun. But with Libra, where we're analyzing 100% of the reads using these novel infrastructures, we can actually have much better resolution for the data set. And all of that, that particular application is also available on iMicrobe. So what's really powerful about iMicrobe is that we're able to integrate not just data that are running on high performance computing through TAC, but we can leverage these really novel architectures such as Hadoop and through this process as well. So coming soon, we are starting to do some work. We realized that trying to move all the data to iMicrobe is absolutely crazy, and that these data sets really live in many different locations. And in particular, there are some amazing resources at EBI Metagenomics and NCBI that we'd like to connect with and connect our community to. And so instead of having all of our users download those data sets to their own CyVerse directories, we want to create the connections that allow them to use these data sets directly through iMicrobe. And what we have done is we've started to work with the syndicate team, which is here at the University of Arizona, and it's led up by Larry Peterson and John Hartman. And what that does is it allows us to have that bridge to these diverse data sets as if they're mounted locally within our server. And the syndicate lets you access data wherever it is stored. So we no longer have to move data sets to use them and analyze them. And so here you can see we're just doing a very simple mount to all of the iMicrobe data sets. This is on our server. And so in just a few clicks of some command line, we have actually mounted those data and it's ready to go. And so what we're trying to do essentially at iMicrobe is develop a resource that is connected in many, many different ways. So we're connecting with CyVerse and the data store to allow our users to have access to large-scale data storage resources at CyVerse, to also access the Texas Advanced Center Computing high-performance compute cluster through the Science APIs. We're developing our own apps and pipelines in iMicrobe, but we're also leveraging those from the community through Bioconda and Biocontainers projects and making them available through our interface at iMicrobe. We're also trying to reach out and connect with the community to share protocols and recipes for using these various applications and really share our know-how in doing this. And then importantly, we're also starting to realize that these data sets are starting to become exceptionally large, and that we don't want to necessarily move them around the world. And using Syndicate, we can connect with these large-scale data sets through iMicrobe. And then lastly, we can analyze these data sets using even really novel compute architectures such as Hadoop. And those resources can also be integrated in iMicrobe. So if you have any questions, we would absolutely love to hear from you. We're on Twitter at iMicrobe, I_microbe is our Twitter line, or you can contact us directly from the site. If you have any issues with the site, we would, or any questions or additions, we'd love to hear from you. And all of this work today was done by just a few very stellar developers. So Ken Ewens-Clark developed the original version of iMicrobe in Perl and Mojolicious. Matt Baumhoff has really, along with Ken, been our data architect for bringing iMicrobe into this Elm-based interface. Joshua Lynch has been really instrumental in helping us bring a lot of these applications, including the biocontainers, to iMicrobe. And then also Il-Yang Choi is a graduate student who works with us who has been developing the algorithms in big data analytics. And I'd also like to say that CyVerse is supported by the National Science Foundation under several grants. And if it wasn't for CyVerse, none of this work would be possible. So thank you very much. And I'll take any questions from this point.
Thank you, Bonnie. There had been some questions in the QA part that I think Ken started to answer, so I don't know if you want to review those. If
Is there a subset of iMicrobe that's concerned with insects? We do have host-associated data, but nothing, I don't believe.
So the question was, do we have any data sets from insects in particular? And as of right now, we have some host-associated data sets, but as far as I know, we do not have data from insects. But we do welcome. We are not. I should say we are not funded to be like a repository for various data sets. But we're hoping that some of the work that we're doing through Syndicate would actually help us connect to those data sets for you from the various locations that they currently live. Likely those data sets have been deposited to NCBI, so you could, in the near future, you should be able to access them in that way. And so you'll see a button coming soon.
So I'm gonna poke in and say that where we're going with the app development, the idea is that you can run the apps on our data sets, but you probably want to run them on your own. And by putting your data sets into the CyVerse data store or putting them on some FTP site that we can download, you could do analysis just on your data, but maybe what you want to know is how does your data compare to what's in iMicrobe. So you could use certain apps like our MASH all versus all, or RepSeq MASH, and then the Libra algorithm all could tell you, for instance, how similar your data sets are to the data sets that we have curated in iMicrobe. So if you have insect-associated metagenomes, you could put it into iMicrobe and then compare it to what we have. Or you could just simply run our tools. For instance, you want to look at taxonomy, you just want to run centrifuge on your data set, or UPROC. We're going to be working on integrating Interpro as well. So while we may not have those data sets, if you have them, you can run analysis on them, analyses.
And I should say where it comes in, where it's most important is if we have the data sets locally, we can index them in that metadata search that I showed you. And we're also thinking now about ways that we can index data sets such as those that are at EBI Metagenomics, because they have nicely curated their data sets. There's a lot of very well-structured ontology and metadata associated with those data sets that we can leverage within our search engine. And so that gives us a lot more ability to really use those different data sets. Any other questions that we had?
There was another question about do we include fungi, protozoa, bacteria, archaea, and viruses? And I said that we basically only have the latter three, but we have been developing some apps around fungi. And as Bonnie just stated, and I also stated in the text, we don't really have the mandate or the funding to bring in new data sets. We pretty much have what Camera had, and then we've added the Hawaii Ocean Time Series and a few other small data sets.
The Ohana Gene
the
Catalog.
Ohana Gene Catalog. But we really haven't been able to curate and add an index, as she is describing, new data sets. But we are absolutely happy to integrate, especially tools that could be used to analyze those organisms, or that would be useful to those communities. I just tweeted about that our first biocontainers-based app is released. It's called Tremgalore. And so there are, I think, 6,000 or 7,000 biocontainers that we could integrate. So now it's just a matter of the manpower to sit down and work those in. Because they've already been containerized, there's a lot less work for us as developers. So if there's one that already exists and it's a high priority for you, let us know, and we'll probably take a few hours and try to integrate it for you. And then it will be available through our app interface, and hopefully that makes it very easy for you to run them and get the results.
Yeah, and I should also say that some of the, we're interested in developing different pipelines that really drive some of the analytics on the site. And so you saw from the webcast that we are annotating all of our data sets for taxonomy for various proteins with KEGG and PFAM. And that kind of information is incredibly useful for trying to understand what, compare and contrast your data with various data sets that you might match to within iMicrobe. And then those pipelines we're making available as apps as well. So what you can do is you can take, you can run those pipelines, run them through your data through the exact, pipeline that we ran, and then your data are much more comparable within, to other data sets within the platform that we have. And we also hope, as I had said, that using your data store, you're able to bring in data sets that way from CyVerse, or you could also, access those data in the near future through these connections with resources such as EBI and CPI. Any other questions that we had?
That's everything.
Okay. Anybody else have anything else in the chat for us?
I don't see anything, Bonnie, so.
Okay.
All right. Well, thank you, everyone. As I mentioned, look in the chat window for the wiki page. I'll also follow up with an email to everybody with that wiki page link, so that in case you didn't capture it in the chat. But thank you so much, Bonnie and team, for presenting iMicrobe and all its really cool features, and shows how CyVerse is the basic platform that then people can use to extend and build and do wonderful things like Bonnie and her team are doing. So thank you very much, and we'll see you at the next webinar.
Okay, thanks. Bye.